All right. Hello and welcome to this exciting showcase session, a wonderful new format that we want to be highly interactive. So with that, I'm now delighted to welcome our first panel member, Billy Lau. Billy is an instructor in the Division of Oncology at Stanford University School of Medicine. His research focus is on leveraging genome technology for cancer detection. Take it away, Billy.
Thanks. Hi, so our group is really interested in using liquid biopsy for longitudinal monitoring of cancer as well as for early cancer detection. Just as a brief review, liquid biopsies are commonly defined as taking a blood draw from a patient and examining the constitutive components inside in order to give either a diagnosis of cancer or any kind of disease condition. In our case, we're specifically interested in looking at methylation marks. Methylation marks are specific base modifications that can exist on cytosines, and these are reflective of the underlying transcriptional programs that are present in cells. So in the top panel, what you see there, you see a normal cell has a specific pattern of 5 MCs in front of a promoter indicating that tumor suppressor gene is, let's say, active. But let's say this pattern changes, this is reflective, could be reflective of a malignancy, and you can see that the tumor suppressor gene could be silenced, and this could be a pattern that you can detect through liquid biopsy research. So in the past year, the thing on the left panel just came out in Genome Medicine a few weeks ago. We've developed a protocol using Oxford nanopore sequencing, whereby we can sequence at very high throughput cell-free DNA using the Oxford nanopore platform on a highly multiplexed fashion. We developed a protocol that increases the throughput by over an order of magnitude, and we can go down to even lower than 1 nanogram to hundreds of picograms of input. This correlates to probably maybe a mil to half a mil plasma. Since then, we've extended this study to look at population cohorts. So on the right is a new study that we're embarking on where we have sequenced a large case control cohort of early stage breast cancer patients as well as negative controls, and also a validation cohort as well for the purpose of early breast cancer detection. To implement this early detection classifier, we employed a couple of different omic modalities. The first is methylation. As we said before, you can train classifiers to look at the aggregate methylation profiles in case and control and compare unknown cases to it in order to derive similarity scores and call whether those could be case or control. Similarly, you can also use alternative modalities such as fragmentomics, whereby the fragmentation patterns of the reads themselves can be indicative of disease states. And this is something that we described in the Genome Medicine paper that we just published. We can use these fragmentation patterns, fit to mixture models, and develop classification schemes as well. Lastly, you can put these two omic modalities together with ensemble learning. And on the right here is some brief preliminary results on a validation cohort showing that we can detect breast cancer at a Newton index of about 70% sensitivity and specificity, but obviously for diagnostics, you can tune it to wherever you want. So that's it, what our lab does. I'd like to thank all these people that are involved in the study and the patients that made the study possible. Thanks.
All right. Thank you very much, Billy. Right on time. Now I would like to welcome our second speaker for this session, Caroline Sauer. Caroline is a postdoctoral research fellow at the Cancer Genomics Group led by Dr. Cortez Siriano at EMBL. So, Caroline?
Thank you, Olivier, and thank you to the entire Nanopore team for having me here today. So for the purpose of this session, I'm going to focus on pediatric cancers and why we think nanopore sequencing of cell-free DNA in pediatric cancers is particularly beneficial. And this is because pediatric cancers are the leading cause of death in the Western world in children or post-infancy. And we know that children, particularly with solid tumors, have very poor outcome and very high relapse rates. However, from genomic studies, we now know that quite a large proportion of pediatric cancer patients have actionable alterations that we might be able to target in the clinic. The key challenge here is to obtain sufficient tumor tissue or material to perform genomic tumor profiling. Surgeries in young children or toddlers is not very straightforward, and even biopsies are not very trivial. So this is where the promise of liquid biopsies come in that I hopefully don't need to introduce you to. So our main research question is, can we use long-read nanopore sequencing to develop multimodal liquid biopsy tests for children with cancer to screen their cancer types and monitor their disease progression? And here's the study overview that we're using. So we're part of the Stratified Medicine Pediatric Study, which is a UK-wide trial or study in which we collect samples, tumor and liquid biopsy samples, from children with solid tumors that relapsed. And we then perform nanopore sequencing as well as Illumina sequencing for validation purposes. And our group in particular is very interested in developing computational tools to then analyze the data, looking at copy number, looking at fragmentomics, looking at methylation. There's a very exciting talk tomorrow by my colleague Hilary talking about her somatic SV caller. And we're also very interested in looking at the methylation signal, as Billy already laid out. This is something that's very exciting, in particular for cell-free DNA analyses. So we want to use the methylation signal to more accurately predict tumor burden, but also perform disease classification analyses. And lastly, we want to integrate all this data to have a nice and accurate way to monitor disease progression and treatment response in children with cancers, and also predict relapse a little earlier. So this is the sort of longitudinal aspect of our work. And just to give you a brief flavor of the three aspects I've just mentioned here, I'm just showing you the very typical cell-free DNA fragmentation size profile that we do get from our nanopore sequencing reads. So you can see mononucleosomal, denucleosomal, and trinucleosomal DNA, but we also get ultra-long cell-free DNA fragments, which are quite interesting. We do detect highly accurate copy number profiles. Here I'm showing an example where we detected clinically relevant focal amplification in ALK and MYC-N. We do look at methylation deconvolution and find that our estimates from the methylation signal strongly correlate with our tumor fractions, and also that cell-free DNA samples from different tumor types cluster separately. So you can use the methylation signal to differentiate between different tumor types. And lastly, here's just one example from one neuroblastoma patient where we had 5 samples that were longitudinally collected, and you can see that the tumor fraction alters over time before surgery, during or after chemotherapy, and also at relapse, with additional copy number events detected towards the end. But I'll finish with thanking all my collaborators, clinical collaborators, all the patients participating in the study, and also in particular Andrew and his team, Nick and Joe, who are right there, who are doing all the sequencing for us. So thank you very much.
Well done.
Thank you, Caroline. A really beautiful example of the value of nanopore and how you can integrate multiple parameters just into one assay, one platform. So with that, our final panel member for this session is Andrew Beggs. Andrew is a professor of cancer genetic and surgery at Birmingham University, the hospital in Birmingham, in the UK. He runs a mixed lab, wet and dry, for oncology, immunogenetics, and also organoid model. Andrew?
Thanks, Olivier. Thanks all for coming to listen to my talk today, and thanks to my lab for sitting there who do all the hard work. Joe and Nick there. I can't take credit for most of this. They're the ones who should be here. I'm going to talk about what we like to specialize in the lab, which is doing things that other people say are impossible. So people say you can't get mutations out of ctDNA on nanopore, and hopefully I'm going to prove to you that you can. So we've developed a homebrew ctDNA sequencing protocol using a multiplex amplicon panel, which we borrowed from a commercial provider, but we've adapted it such that you can take the amplicons and feed them straight into an LSK114 kit. The reason we've done this is because the current methods for ctDNA analysis, hybridization, are slow, and so by using PCR, we can very rapidly go from a sample to a result within about 8 hours. We've also made it work on the flongle, and it costs £75 all in, including all the enzymes, reagents, and sample input. And you can show it also works between anything from 5 nanograms and beyond of both FFP and ctDNA. And the protocol generates sufficient coverage of 10,000x in 8 hours. So this is a truly patient-facing test that we can very rapidly take into clinical use. Now, they said you can't detect low variant allele frequency with nanopore, so this is IGV for those who haven't seen it before, and the top panel is non-duplex, but the bottom panel there is duplex sequencing to show you the power that duplex sequencing adds to these amplicon runs. And this is KRAS codon 12 and 13, which is one of the most commonly mutated oncogenes. And in this reference sample we use, with another variant allele frequency about 15 and 6%, you can see that both the duplex and the non-duplex detect it. And what you can hopefully notice, and bear in mind this is a very low input sample, is how noise-free the reads are, how easy it is to pick out the mutations, showing that this panel really is the answer to these things. Now, the next problem is that with these circulating tumor DNA panels, it can be very difficult to detect long indels. And this is where nanopore comes into itself. This is a very long deletion, 15 base pairs in EGFR, a common one in lung cancer at 2% variant allele frequency. Now, in the top panel, you can see without duplex on, it sort of works, but it's a bit noisy. But you can see at the bottom with the IGV panel there, it displays the indel there, clear as day, easily pickable out, and it can easily be detected with a variant caller such as Claire3, which we've been using. Final one, PIK3CA, E45K, another common variant that you detect in lung cancer and circulating tumor DNA. Once again, 1.5 variant allele frequency, very easily pickable outable. You can see at the top there, it's relatively noisy, but duplex removes all the noise. So we've shown now with this panel, which can take commercial PCR panels, modify them and put them into nanopore, we can get a variant allele frequency of down to about 0.1 to 0.2%, which is a really transformative technology for circulating tumor DNA analysis. So when we compared it to a reference panel, we detected 95% of the mutations in the Seracare ctDNA reference panel that were covered by our panel. The only one that wasn't detected was one with a 1% variant allele frequency, and we think this has nothing to do with the panel. We did detect it. It's to do with the tweaks we need to make to the duplex caller. Thank you very much.
Thank you, Andrew. And actually, thank you to all three of you because they've been sharing pre-publication data. So a lot of it was really a preview for many people, so it's very kind. So now the goal is really to have a discussion at high level, and because a lot of it is not published yet, we will wait for the publication to have all of the technical details. But do you guys want to share anything on that point before we open to the more general question? No?
It's a secret. It's not. I mean, it's not a secret from the panel from our point of view is you can take any commercially available PCR panel. Well, I'm not going to name the manufacturer we've taken one from, but you do need to fiddle around with the polymerase conditions. Say Joe and Nick have done all the hard work. Need to fiddle around with the polymerase conditions to optimize the amplicons. But then once the advantage then is the company has done the balancing for you, so you can then take that PCR panel and stick it straight into the flongle. It's not that complicated. I shouldn't really say that. I want you all to work with me. But actually, as a technique, it's very straightforward. And you'll see it in the paper.
Thanks, Andrew. Do we have questions from the audience?
Hi, Luca Mazzarella from Milan. So regarding the last talk, so it's not entirely clear to me, however, what is the advantage of sequencing the PCR amplified panels with nanopore as opposed to, whichever other short read sequence? Because your amplicons are small in size, aren't they?
Yeah, I mean, our point is that, so we've tried we do quite a lot of stuff in our lab. We do the TrueSight Liquid 500, another hybridization panel, and the workflow to go from a hybridization panel to result is 5 to 7 days, which is too slow in some of these cases. One of the things that hopefully I perhaps didn't make it as well as right at the beginning is that we can go from the DNA to the result in 8 hours, which is important. You can't achieve that with hybridization. And you can't do that with short read based sequences because you need to run the flow cell for 24, 36 hours to get data. So that's the key advantage to us, and that's why we developed it.
Thanks.
Thanks. We had a question from Anna on the first row.
Thank you for introducing your different methods. Great to see the different modalities being used. When we think about liquid biopsy, we're thinking early detection. Early detection means difficulties with, actually picking up those rare mutations just by default because they're early. And I think this is what we're seeing with current methods that really, okay, they can detect those variants, but they're actually not detecting them early enough at the earlier stages of cancer development. How are you trying to approach that main issue from my perspective with your current methods that you're developing? And that's the question to the whole panel.
I mean, I think in general, the hardest problem really is getting enough DNA for sequencing. So in our case, we've demonstrated that we can start with actually a very small amount of DNA, but the ideal method would be processing a large volume of plasma, get as many DNA molecules as possible, and getting as much throughput. That's more or less a budgeting issue then, right? What is the trade-off between how many reads you need for specific sensitivity and specificity, and how much of a cost analysis that you're willing to put in? Because clearly you can sequence, if you have a lot of DNA from, let's say, 10 mils of plasma, 20 mils of plasma, that's like 2 or 3 blood collection tubes. You can get a lot of DNA, and you can sequence that to 30x on a couple of Promethean flow cells. But the question is, do you really want to do that?
So
Caroline, do you have something to add?
I do. From my point of view, so I agree with Billy on the more plasma we have, the more DNA we have, the more likely it is that we're going to detect the mutations that are relevant or of interest. In the case of pediatric cancers, that is obviously not always the case because the plasma volume, the blood volume that we get is very, very small. So we're kind of sort of scratching at a limit of what we can detect. But I think there is a lot of value in being able to read out the methylation data that might be more informative on a read-based level than picking out particular mutations might be. And what we're trying to do at the moment, so obviously we do long-term want to move towards early detection, but another application that is often forgotten about when we talk about early detection is also the disease monitoring and earlier detection of relapse. And I think that's in particular where the application of nanopore sequencing of cell-free DNA is going to be quite strong and maybe easier to achieve in the nearer future before we then go to true early detection and solving it all. So that's my point of view on this.
Andrew?
I think one of the things we've done up until recently is always relied on mutations to tell us whether somebody's got cancer or not. It's not that easy. You know, the more, like Carolyn and Billy have both said, the more plasma you have, the more likely you are to detect them. But what you do notice is a shift in tissue type or tissue phenotype, which can only really be detected epigenetically. So the methods that Billy and Carolyn and we use to detect epigenetic change are probably going to be more important for these early screening assays. So somebody asked about input, like Carolyn says, they use a mil of plasma from the SMPs, and then we're talking about 3 or 4 nanograms of DNA. But one of the things that not just us, but others like Stanford realize is the nanopore kit, it works on femtomolar loading, not quantity. So if the DNA fragments are so short, 150 femtomolar of DNA is, 5 or 10 nanograms of DNA. So that's easily within the reach of most of these pediatric samples. And that's the key to unlocking a lot of these epigenetics tests.
So Billy, on the very low input, do you have some specific comment? I mean, from the preprint, how low you went into quantity of material?
Yeah, so I mean, in the previous published paper and also in the current study that we're doing, we work with a very small amount of plasma, about like 0.5 ml of plasma effectively, that goes forward to sequencing. We did a couple of measurements about what that roughly corresponds to. It's anywhere between, let's say, a couple hundred of picograms of DNA up to like a little bit over 1 nanogram of DNA on average. And that's more than enough for us. But the key trick is that you have to have a very optimized library prep.
Right.
And there's a couple of tricks that we use. It's not that interesting. You just read the paper. It's more about like being very smart about how you make a library, monitoring what your conversion efficiency is. And that's one thing that we're able to do very well that we were able to monitor in the genomedicine paper. And then once you have that, then it's more about productionizing the whole process, because a lot of the follow-up study that we have in our breast cancer cohort, it's about sequencing large numbers of patients. And in that case, you have to have some automation, some good hands, like a good extraction system to do this in volume. That's the hard part.
Thank you. Do we have comments specifically on the early detection before we move on to the next topic? Comments or opinion from the audience? Observations? Specifically? Go ahead.
So I have a question about the DNA methylation. So I guess this is for Carolyn and Billy. Billy, on one of your slides, I think I saw it said that you're querying 400,000 CPGs. So the question is, what is the optimal number of CPGs that you want to be looking at, and what depth of coverage is optimal?
Yeah, so the 400,000 CPGs refers to our feature selection step, where we have a case control sample. We have all the CPGs that were sequenced, and we look for statistically significant differences in the methylation values between our case control cohort, and what falls out is the 400K CPGs. But obviously that changes with the size of your cohort, how deep you sequence and everything. So for us, it's not a specific cutoff, but more to do with after you do your sequencing, you do a statistical test, do the FDR correction, and whatever comes out is what you're looking at for feature selection.
But it's whole genome, and
it's
the
whole genome,
400,000 come out of the analysis.
Yeah, that's correct.
Okay. And what depth of coverage?
We have roughly like a couple million reads per sample, so that roughly translates to also a couple million CPG sites per sample as well.
So, I mean, we have done this as well, and I would add that, like Billy says, there's no optimum amount. So we can see as many as 1 million CPGs in some samples, and some have only got 50,000. Generally, 5 nanograms of DNA will give you about a gigabase, 2 gigabases of sequence data, perhaps more, depending on the sample. So, I understand from the work that Carolyn and Isidro have done that, the classifier doesn't need that much information to pull out methylation data. So it sounds once again a bit flippant, but you literally just put in what you've got and see what you get out, and usually that's enough to classify. It certainly saves taking the gallons of plasma off children that used to happen.
Yeah.
Cool. Jeroen?
Yes. Thanks a lot, guys. Really awesome. So comparatively to the mutations and the methylation, how important is the fragmentation pattern and you look at size, I presume. But do you also, for instance, look at the end motifs of the fragments that you
Yeah,
observe?
so I don't know, do you want to start or?
Yeah, so your question is like, I think two parts. One is looking at the methylation and also the end motifs, right? So the end motifs we haven't looked at yet. That's a really interesting question. But could be some biases depending on what the end repair step cuts off. So that's more of an open thing. In terms of methylation profile, I think that's, in general for me, in my opinion, that can be more informative than just looking at the specific cancer mutations, because methylation is reflective of overall transcriptional programs in a cell, right? That can happen on multiple gene levels and whatever. And so methylation patterns are generally going to be correlated all across the genome. So in general, when you're looking at, let's say, one SNP, let's say KRAS G12D or something like that, or BRAF V600E mutation, that's at one spot. And you want to look at that one spot, that's really hard to do, right? If you only have like 1 nanogram DNA, there's 150 molecules at that one spot that can give you that information. Versus if you look at like CpG sites, there's millions of them that could be correlated with each other. So it's much more informative to do that, as opposed to just looking at SNPs. But that's my view.
Yeah, I pretty much agree. And we do look at the size profiles as well, as you saw on the plot earlier. So I think it's also a very important signal you can do in silico size selection and other things that will be very informative, enrich your sequencing data for putative cancer reads, which will be very helpful also for methylation analyses. And of course, there's a lot of work being done on nucleosomal footprinting and trying to infer gene expression. And what we're interested in is then also understanding how this correlates with the methylation signal that we read out from the same data, and expect that it should correlate because it's about gene expression. About the motifs, I think this is going to be quite challenging, because what we know so far about the fragment end motifs is from Illumina sequencing data, and the technology is obviously very different to nanopore sequencing data. And the challenge that we're going to have with the nanopore sequencing data, so there's no doubt that we're going to find different motifs in the cancer reads compared to the normal reads, but I think those motifs are going to look very different to the Illumina sequencing motifs that we know. So we have to redefine what we consider cancer-specific fragment end motifs. And the reason for that is that, especially when you start sequencing the DNA, the first few bases are a bit inaccurate, and then you massively depend on the performance of the base caller to remove the adapter sequence. And if you obviously remove the adapter sequence just one base pair too many or one base pair too little, you're just looking at the last 4 base pairs of that fragment, and that's going to change the motifs. So obviously Illumina has other limitations, but I think it's going to be very interesting but quite challenging and not easily comparable to the data that we might already know from previous publications using conventional sequencing approaches. But it's definitely something that we're looking at.
Andrew, any comments on the jagged ends?
It's been so expertly answered. What else could I say? The only other thing I would add about fragment analysis is, there were recent papers on pregnant women as well that showed it's not just about the 175, 300 base pairs. We are detecting long fragments now, and that's the other advantage long read sequencing gives you. I mean, we routinely, in the data we generate for Carolyn and Isidro's group, 1,000 base pair fragments. They do exist in plasma circulating from tumours, and they are tumour-specific because they're methylated in a tumour-specific way. So these long reads are actually quite informative when it comes to classification, and I think they'll play a more important role and a unique role.
Yeah.
Thanks. We have about a couple minutes left, so do we have any further questions? Yeah?
Hi, it's quite a technical question for Billy, is that okay?
Yeah.
Yep.
If it's okay for him, yeah?
Yeah, so love your work, love the paper, and particularly love how much detail you've given in the methods. So I work at the vet school up in Edinburgh, and we've been inspired to kind of try out and develop a liquid biopsy for dogs. So do you have any tips or tricks for getting started, particularly as we might move towards kind of kit 14 versus the older kit that you used?
Yeah, so we're actually doing kit 14 on some other studies in the lab right now. I would say if you read the paper, there was a second end repair and A-tailing step. You don't need that anymore, and that's it. Then you can do it on V14.
Thank you.
We do have a technical suggestion as well. I can't say it out loud. Joe knows what it is. There's a certain brand of Eppendorf tube that you shouldn't put. It's not made by Eppendorf. Do not put your plasma in it because it destroys the nanopore. But come and talk to us afterwards. We can't mention it in public because we'll get sued.
All right, thank you very much. So we are running out of time. That being said, you've seen them. They're going to be around. So feel free to walk out to Andrew, Caroline, and Billy. Let's thank them one last time. That was a really great session.